Good afternoon. Welcome to all of you here at Lawrence Berkeley National Laboratory and all of our colleagues who are viewing at parties around the globe. Welcome to the 2019 Berkeley Lab Research SLAM! I'm David Gilbert from the DOE Joint Genome Institute, soon to be your neighbor just down the hill here. And I really appreciate you having me back again to host, second year in a row. Hope I don't disappoint. But one condition I made to be here today was I didn't want to be hung out to dry and be up here alone. So I thought, who best to serve as my co-host? I thought about this long and hard, but I came up with the perfect solution. So I give to you the reigning 2018 Berkeley Lab SLAM champion, Sinead Griffin! Sinead Griffin, everybody! Hey, Sinead, what's up?
Hey, David, how's it going?
Oh, pretty good. What have you been up to this last year since you're in your championship reign?
Well, a bit of gloating and telling everyone a lot about dark matter.
Oh, yeah, everybody knows what dark matter is, right?
Right? Actually, I think we should check. So, hands up if you know what dark matter is. A lot of people. Trick. That was a trick question. No one knows what dark matter is.
Ain't that the truth? And yet, that's how you won the big bucks last year on Dark Matters, as I recall was the name of your presentation. So with all that money in the bank now, I guess that affords you the leisure time to be here today on stage with me as my co-masters or mistress of ceremonies, MC, right? So what exactly was your motivation to join me here in this auspicious place?
Well, you wanted a co-MC, so I really couldn't pass up the opportunity to be your MC squared.
Oh. I'm sorry, that comment went over my head at the speed of light. But I have a complication for you, Sinead. We have another MC in the house, Mary Catherine O'Connor, right? Go by MC. So how does that affect your equation? MC to the third? What's up with that?
Sure. So speed of light being the electron mass, then you got like 2.455 by 10 to the minus 5 kilogram per meter cubed centimeter cubed or second cubed. Is that right?
Oh, yeah, everybody knows that. Give it up for one of the brightest stars in the Berkeley Lab constellation, Sinead Griffin, everybody. And on that note, least we forget, our safety minute. Is somebody going to time me, my safety minute? All right, here we go. Do we know what to do in case of emergency? Of course we do. We work at Berkeley Lab, one of the safest places on earth. Hey, Carrie, what's going on? So head out and head into H1 lot. If it's overcrowded, H2's out there. It's a pretty good alternative. If you see that I'm in distress and maybe need defibrillation, just around the corner at the exit here, you'll find the paddles. And anybody know how to use those things? Thank you, Kathy. I appreciate that. All right. Who here is going to call 911 if something bad happens? Anybody? Volunteers? Thanks, lady. All right. Who will lead the evacuation? Russell, thank you. All right. Who will meet the emergency vehicles? Oh, hey, Kia. Thank you for that. All right, one more thing. Who's going to sweep the room? This is a dangerous position, but somebody's got it. Mike, are you up for this? Sweeping the room? Anybody? Sweep the room? Oh. That's demerits off your grade today. I will sweep the room. I'll go down with the ship. All right, then. Sinead, please.
Yes, I have a big job tonight. So I have to actually present to you the one person. Oops. You didn't see that, right? Close your eyes, everyone. We're back. Okay, you didn't see that. Go backwards in time. Okay, so I have the privilege to present to you what we believe to be the postdoc's biggest fan, because he said so on Twitter. He said that Berkeley Lab postdocs are the best in the world. Here, paraphrasing his tweet. So I give to you Director Mike Witherell.
Hello, and you are the best. And I'm not running for office. But I do, every time I talk about this laboratory, everybody comes in, I emphasize what a crucial role the postdocs have in how we get our work done. I think they are the key to a successful research institution anywhere. I also point out that it's the last time that you'll be able to spend all your time on your research, because everything you do after this, you're going to have less and less time for it. So even though it doesn't seem, oh, okay. Sorry. Okay. So you are the best, and but it's looking back on it, you will remember this as the time that you could actually spend as much time on as your research. As I'm looking at all these people like David here in the front row, and that he wishes he had that much time to spend on his research. Once you get into management or faculty, it changes. So it's actually a best time of your career in many ways. So let me say I'm thrilled to have this level of excitement. I will also say that there's no way you could have gotten me to do this when I was a postdoc. I was hopeless at public communication. So you're way ahead of the game already. So looking forward to it, and have a great time, and I look forward to see who's going to win. So have a good time. Thank you.
Thank you, Mike. Give it up for our director, Mike Witherell, please. If you're not dialed in already, dialed in doesn't work really in this sense anymore, does it? Here are the details on our hashtags and handles, except for one's missing there at least, at JGI. Can't help it, sorry.
That's cheating.
Well, it's the privilege of the co-MC, what can I say? That's it.
Yeah.
And on to you.
Okay, we're nearly ready. So this is the boring stuff. Everyone has their phone off, right? You're only allowed to use your phone if you're going to tweet. If you need to leave, we've got a very conveniently placed round of applause in between each person. Please do not come back into the auditorium if it's quiet. So please wait until you hear that round of applause so you're not interrupting the slammers when they're giving their presentations. And one more thing, each of the slammers has been tasked with spending just 3 minutes on their slam. Now, if they go over time, it's going to be your responsibility to stop them, and you will see 2 different signals. So just before they're supposed to finish, they will see this green signal, which means they have to finish up. And if they see this red signal, and if you see this red signal, it means you need to start cheering and screaming and clapping so that they stop. So let's just do a practice run. Here we go. Okay, you got that bit down. Okay, finally we have 12 finalists. Does everyone here have a program? Yeah, everyone has the program. So if you flick to the back page, you will see all of the entries there and a nice perforated edge. So once you've listened to all 12, please don't vote until you've listened to all 12 slammers, you can pick your favorite, and there will be collection boxes on the way out so you can vote for your very favorite. The finalist will be, or the winner will be announced at 4:30 at the cafeteria, so please make sure to vote. And there will be directions at the end on who to follow if you can't find the cafeteria on time. Okay.
And for those of you who are feeling left out of the process, you too can play along, except for those at home. You'll just have to bask in the glory of those who win the trivia contest. So in between the speakers, as a certain intermezzo, we'll be asking questions, and you will be answering them. And we will have the privilege of picking the distinguished party who seeks to answer them. So better get it right. A runner will go up to a mic, wait for the mic. Everybody in the world is waiting to hear your answer. So no pressure there, okay? And then if you answer correctly, a lavish prize will be in store for you. And sorry about those at home and around the world, because you'll just have to bask in the glory of the others, kind of the opposite of Schadenfreude. So what else we got here?
Well, very important thing.
That's the judges. Tell us about the judges, Sinead.
So we have 6 very esteemed judges here today. Could all the judges actually put up their hands so we know who to bribe?
Don't be shy.
Right here along the front row. So we have Faith Jukes from Berkeley Lab. We have Patricia Falcone from Livermore, Vivette Lane Newton from UC Berkeley, MC O'Connor, Rigel Robinson, Berkeley Council member, and Dee Rossiter. So thank you very much to the judges. You've got a hard job today, but thank you for joining us here for the SLAM.
Bravo. All right.
It's almost time.
Now the thing you've been waiting for. Something you've been waiting for. So let's get this 2019 Berkeley Lab Research SLAM party started. Come on! First up, hailing from the biosciences area, the DOE Joint Genome Institute. Please welcome to the stage Sarah Calhoun.
Alternative sources of energy are critical for the long-term survival of our planet. Last year, over 140 billion gallons of gasoline were consumed in the United States, and most of our gas comes from fossil fuels. But there's potential in a new alternative fuel source that comes from a group of microscopic organisms that perform amazing chemistry. This group of organisms are the microalgae. I'll give 3 reasons why microalgae make a great source for biofuels. First, they can perform photosynthesis. They take up carbon dioxide from the atmosphere, and with water, convert sunlight into chemical energy. This means they're self-sustaining and carbon neutral. Second, they naturally produce molecules that are precursors to fuel. They accumulate these oily droplets, which are made up of a class of molecules we call lipids. These lipids can be extracted and then converted to biofuels. And third, many species of algae are aquatic. We can cultivate them in water, including saltwater or even wastewater, rather than using land, which could be used for growing food instead. This sounds great, right? But the problem is the overall cost of making algae-based biofuels is still too high to compete with conventional fuels. So we're still looking for the right strain of algae that will give us the most bang for our buck, the fastest growing, most efficient strain. Our collaborators across different national laboratories are screening for which strains of algae grow the best over a variety of conditions. Oftentimes, the most promising strains are new to us, so we know very little about their biology. The goal of my research is to characterize these new strains of algae. At the Joint Genome Institute, where I work, we sequence the genomes of candidate biofuel strains. The genomes act as a blueprint for those who perform the genetic engineering. We also evaluate how the algae respond when we change its growing environment. For example, we lowered the growth temperature and then saw different genes turning on in response. This helps us understand how the genes are functioning in the cell. From this work, I predicted several genes that could be involved in affecting the growth rate. And now the genetic engineers are testing these genes. Our work on the genomics is helping the genetic engineers create better strains. And better strains means higher production and overall lower cost. Each day, we're making steps towards the commercialization of algae-based biofuels. And I'm optimistic that someday soon, you will see algae-based biofuels at the gas pump. Thank you.
Okay, trivia time. Are you all ready? Are you all ready? Yeah. That's better. Okay, first up, which Berkeley Lab Nobel laureate can you find pictured next to Shirley Temple hanging in building 70A? Oh, we have one person and we have another person, so we'll try here first.
Kathy Borns.
Glenn Seaborg.
Say again, please.
Glenn Seaborg.
And the answer is?
Glenn Seaborg.
Incredible. Congratulations, Kathy.
All right, up next, we have, all the way from the Earth and Environmental Sciences area, it's Megan Dillon.
If you imagine the icy, desolate Arctic tundra, you might not picture living organisms hibernating underground. These creatures have been sleeping in the frozen Arctic soils for thousands of years, but now climate change might wake them up. In addition to these slumbering creatures, frozen Arctic soils contain about 1,300 trillion kilograms of carbon, which is about twice what's already in the atmosphere. While the soils are frozen, that carbon stays where it belongs, underground. But as temperatures rise, that carbon can get released as greenhouse gas. When greenhouse gas is released from the soil, the temperatures rise even further in a climate change feedback loop. People fighting climate change often overlook the enormity of this feedback loop because the organisms that are directly responsible for it are too small to see with the naked eye. Single-celled microorganisms wake up when the temperatures rise and start to eat the carbon that's been stored in the soils for millennia. There are millions of species of microorganisms in just 1 gram of soil. The species that are awake while the soils are frozen eat the carbon, metabolize it, and release it as greenhouse gas slowly. But the microorganisms that wake up as the temperatures rise eat the carbon, metabolize it, and release CO2 and methane much more quickly. Right now, we don't have enough information about these microorganisms to include them in climate models, which is a problem because that data is crucial for better predicting climate scenarios in the coming decades. To provide the information about microorganisms and their digestive issues to climate scientists, our lab has taken 3-foot-long cores of soil from several sites across Alaska and brought them to Berkeley Lab to study them. We've hooked them up to many different kinds of sensors to measure differences in the soils at these different locations. We're heating them up very carefully to mimic the natural thaw conditions. As the soils thaw in the lab, we're collecting the microorganisms, their DNA, and the gases that they release. We're learning all sorts of amazing things about the diversity and metabolism of these microscopic communities. But most importantly, we're trying to figure out what slows them down. If we can understand what lets the microorganisms keep sleeping, then one of the scientists fighting climate change might be able to figure out how to keep more of that 1,300 trillion kilograms of carbon where it belongs, underground. Thank you.
Ready for trivia time? All right then. Brace yourselves. In 1946, lab mechanical engineer Cyril Orley was asked to design a symbol to indicate the presence of radiation. What colors, and this has to be exact, we need to know the colors used for this symbol. Go!
We have someone at the back. Hold on.
No cheating.
What do you think?
Pink and turquoise blue.
Repeat.
Magenta pink, or like a pinkish, and turquoise blue.
Judges? Dope. Gee, close enough? Close enough! Close enough, everybody. That's the nature of science. It's just close enough, right? That's how it goes. All right. Now, from the formidable energy technologies area, I present to you Michael Gerhard.
I want you to think about how you got here today. If you're like me, you drove, or maybe you took the bus, or you biked, or you walked. The problem with a lot of these modes of transportation is that they rely on burning gasoline in an engine to move a vehicle forward. This process releases carbon dioxide, a greenhouse gas that contributes to climate change and global warming. In fact, about a pound of carbon dioxide is emitted for every mile that we drive. So imagine you're like cruising down the freeway, and every couple of minutes you take one of those 5-pound bags of flour and just chuck it out the window behind you. But this flour also melts the ice caps. Now, it doesn't have to be this way. We have the technology today to power your car using hydrogen gas, using a device called a fuel cell to extract electrical energy from a chemical reaction between hydrogen gas and oxygen in the air we breathe. And we can do this all for the low, low price of $58,000. That's more expensive than a Tesla. Now, there's some advantages. Fuel cell cars, you can refuel in about 5 minutes. Batteries take a couple of hours to fully charge. And the 300-mile driving range of a Toyota Mirai is a lot longer than all but the best long-range Teslas. But still, 58 grand is a pretty hefty price to pay. Now, one of the reasons these things cost so much is we actually have to oversize the cells and use expensive materials to get enough power to push the car forward. Here at Berkeley Lab, I do computer simulations to understand what limits the performance of these cells and how we can optimize them. And we found that water in particular plays a very important role. In the middle of every fuel cell is a thin plastic sheet called a membrane that keeps the hydrogen and oxygen gas separate and allows us to control the reaction. The membrane needs to stay wet to do its job. If it dries out, the cell dies of thirst. But we can't let too much water accumulate in the cell either. If it does, the cell floods, and the hydrogen and oxygen can't get to where they need to go, and the cell essentially drowns. My simulations predict how much water is too much, how little is too little, and where in the cell it's produced, so that we can test different water management strategies and optimize their performance. By doing this, we can get more horsepower with fewer cells. We can also test new, inexpensive membrane materials that might absorb or release water a little bit differently. This will allow us to bring the cost of fuel cell cars down and put more of them on the road. And if we're successful, we'll be able to stop throwing trash out our window whenever we drive, and we can slow or even stop the harmful effects of global warming.
You got some fans there in the audience too. Okay, another trivia round. Prior to the new IGB building, what large piece of equipment did the prior building 51 house? And here's a hint, it's called a something lot. We've got someone up there. So what was there before the new IGB building? And we've got someone at the back as well. Green t-shirt first.
Do we
I
allow
think.
ringers?
Oh, up here. Dave. Oh.
You there. Where?
The Bevatron.
Is the Bevatron right? Does anyone else know if the Bevatron's right? Yeah, it's the Bevatron. Good job.
Bravo.
And that's your new home, David, right?
Not the Bevatron, I hope.
It's got high energy too. All right, next up, we have, all the way from the biosciences area, it's Lauren J. Bush.
Who here has ever rented or bought a place to live? So you know that the three most important things about finding a new place to live is location, location, and say it with me, location. This holds true for bacteria, which is the focus of my work. I study soil, particularly soil bacteria that interacts with the roots of plants, and I study it in a new way that I believe can tell us more about these interactions. The conventional method to study these bacteria is to grind and find. We literally throw the entire root ecosystem into a blender and then work backwards to be able to find bacteria's relationship to the plant that it lived on. This is limiting because we don't understand where and how that bacteria lived on the root. In other words, scientists have not quite figured out how bacteria finds its home with location in consideration. And I want to change that. 3 grams of soil contain as many bacteria cells as there are people in America. And just like you, each one has their own particular housing needs. Some like to snuggle up next to the root of a plant, forming symbiotic relationships that allow both to thrive. Others like to live far away from plants, scavenging for survival. I've noticed that there are many external impacts on the livelihoods of just one bacteria. As examples, my bacteria will change its neighborhood on the root depending on how much water the plant receives or the age of the plant. How do I know about the dynamics of this bacteria? I've developed a device called an imaging ecological fabrication, or imaging ecofab. The imaging ecofab can support a small plant and its surrounding bacteria for its entire life cycle. Excitingly, this device can be placed on a high-resolution microscope, allowing us to see all of the nuances of the root structure and the different places that bacteria could call home. We pair the images that I take with genetic and chemical analyses that allow us to know both who is in the neighborhood and what they like to eat. We combine all of these pieces of information to paint a full picture of the bacteria's diverse lifestyle. Now, why is it important for us to understand what a bacteria wants in a good home? Well, we've increased global food production using methods like domestication and chemical fertilizers and modern plant breeding, but we still have hungry people. Adding beneficial bacteria to farm soils is the next wave of innovation for us to feed ourselves. We need to understand where and how these bacteria interact with crop plants in order to capture their benefits on our farms. My innovations will define where and how these bacteria live on the plants, driving our next wave of innovation. Whether they are looking for a cozy apartment or a fixer-upper, I believe that I can find bacteria at the root of their dreams. All I need to do is find bacteria's ideal location, and say it with me, location. Thank you.
Next up, From the Earth and Environmental Sciences area.
Trivia.
Oh, right. It was so trivial, I forgot about it.
Wah, wah.
All right, brace yourselves. Speaking of the Bevatron, how many Nobel Prizes were awarded for the work done in whole or in part at the mighty Bevatron?
Oh, we have someone right where I can't get him. It seems.
Hey, Don, you're going to have to tell us each one of the winners of those Nobel Prizes. It's only fair.
All right. So, okay, you're going to have to meet me halfway, I think.
It's
Four.
a four?
Four.
Four? Are we feeling lucky? What do you think? Yay or nay? Yeah. The answer is 4 Nobel Prizes. Next time you're truly going to have to recite the wise decision. All right. After so much suspense, from the Earth and Environmental Sciences area, please give it up for Faji Main.
Can you imagine a single day without water? In Niger, my home country in Africa, people struggle every single day to have a glass of water. Water is a human right. California, our home, is already experiencing climate change, the most critical problem of our society. You guys certainly remember how devastating the wildfires were last November, or how ugly the extreme events were over the past years. And this is just the beginning of a new era. Severe droughts, unprecedented wildfires, extreme precipitations. How does this new normal affect our water resources? Water is a result of several very complex processes, present in different forms: liquid, gas, ice. Water interacts with the atmosphere, the Earth's surface, and the underground. It's extremely difficult to capture water movement across the Earth because it can change speed, position, and form at the same time. We reproduce this movement with mathematical models. The future is highly uncertain and complex, and we need state-of-art physics methods to tackle it. Unfortunately, this model takes so much time to capture water's journey across the Earth, but we don't have that time because it's urgent to make decisions. Thanks to advances in high-performance computing, we are able to capture water movement across the Earth in high resolution, on a regional scale, and in near real time. To check if our model is able to reproduce the water movement across the Earth and the real life, we compare them with measurements performed on Earth, but also in space by remote sensing techniques. Our results show that, as humankind, we are our own enemies because our excessive use of water has more impact on water resources than the whims of Mother Nature. On the other hand, our study found that the devastating wildfires occurring in California can actually contribute to increased snowpack, the main source of water supply in California, but also river and groundwater storages. As scientists, we love curiosity and challenges. As human beings, assuring that the future will be bright for the next generation is our core value. And never forget that the next generation, whether they are from California or Africa, needs water. Thank you.
All right, trivia again. So, in the 50s, this Berkeley Lab scientist conducted important research on medical imaging that led to the development of the scintillation camera. Anyone? Oh, we have. I think that's slightly cheating if the director. But anyway.
Harold Anger.
How angry was he?
He was very angry. It was
Harold
anger.
Hal Anger. Congratulations, James Simons.
And our next slammer, coming from the energy technologies area, is Akansha Menon.
We all think of oil and gas companies as energy companies. But what if I told you that their biggest product is water, or wastewater, to be more precise? This is because the production of 1 barrel of oil requires over 10 barrels of water. And given that these companies produce 1 million barrels of oil in a single day, you can just imagine the scale of the wastewater challenge. Since this water is too contaminated to reuse, it's disposed by injecting in deep wells underground. Last year, scientists conclusively showed that deep well injection was causing seismic activity in regions with no earthquake history, like Oklahoma, for example. So all of you may be wondering, instead of disposing this wastewater, can we purify it to meet the growing demand for clean water? The answer is yes, and this is the focus of my research, where I'm developing a desalination technology to separate salts and contaminants from the water. There's thermal desalination, where you use huge amounts of heat to evaporate water, leaving the salts behind. And then there's membrane desalination, where you use large amounts of electricity to force water across a selective membrane that blocks the salt out. This is the crux of the problem. Desalination is very energy intensive, especially when we have to do it on such a large scale. To address this, in my research, I'm developing a hybrid membrane and thermal desalination system that's powered by solar energy. Typically, when we think of solar, we think of PV panels that convert sunlight into electricity. In my research, instead, I'm converting sunlight into infrared heat. Our secret sauce is a temperature-responsive material that performs 2 very critical functions. It draws water across the membrane, separating it from the salts, and then itself separates from the water by absorbing this infrared heat. This process requires virtually no electricity, and the thermal energy consumed is 1,000 times lower than evaporating water. I'm very excited to share with all of you that we have successfully demonstrated this in the lab, and we will be working with our industrial partners here in California to field test the technology. Thus, by converting this waste into a resource, we can close the water loop, recycle water for agriculture and potable reuse just by harnessing the power of our sun. Thank you.
Trivia time, people. This element, discovered at Berkeley Lab, is widely used in smoke detectors.
First hand up was over here. Oh, I'll go around this way.
The tension mounts. Does he have the right stuff?
What is americium?
And the answer is nothing. Americium, everybody! Next up from the mighty computational sciences or computing sciences area, give it up for Bashir Mohammed.
Seven point one. On Friday, the 5th of July, 2019, the U.S. Geological Survey, USGS, reported an earthquake of magnitude 7.1 in Southern California. The USGS has about 2,000 sensors used to monitor earthquake in the US alone. You see, these sensors are connected to a network, and they transmit traffic in form of sensor data in real time. So imagine a scenario where there is traffic congestion on a network, and these readings do not get to the USGS on time, and they are unable to give timely warnings of the earthquake. Imagine the devastation to human life. And no, before you ask me, let me tell you now, my research is not about earthquake at all. The primary aim of my research is focused on developing artificial intelligence algorithms to control high-speed networks in high-speed network towards controlling airport traffic congestion. Today, we live in an interconnected world which is heavily dependent on the internet, which means network traffic will continue to grow exponentially day by day. Picture this. Imagine waking up in the morning and you cannot connect to the internet with your cell phone or tablet or even PC. Why? Because the network is congested or down. OMG. Now here's the catch. What if I tell you that there's a lot of research that has been done in the area of network traffic congestion management? But there's a problem. Most of them have looked at the issue from a reactive point of view. Basically, they've tried to address the traffic congestion after the traffic congestion has occurred. This is clearly not going to be easy, and that's why I come in. So my team and I have utilized the concept of basic statistics to analyze and explore this massive amount of network data, plus the power of advanced computer simulation and artificial intelligence to predict real-time network behavior and the next level of traffic congestion. In simple terms, we have used historic traffic timestamp data to predict future network pattern. This, we believe, will provide network engineers with prior information of the network behavior, which will help them to be able to avoid data traffic congestion, network downtime, and above all, save DOE millions of dollars on equipment cost. And if this is successful, it will indeed answer what is arguably one of the most challenging open research questions in energy science network. Thank you.
It's trivia time again, OMG. I stole that one. What was the first element Berkeley Lab played a role in discovering? Oh, we have someone right at the back. You can just guess a number. I mean, there's only so many elements.
I'm going to guess berkelium.
No? So someone else has a suggestion over here? Up at the back. Yeah, we have someone at the back all the way over here. You could have made it easier.
Get my steps in. All right, who is it? Margie? Don't get it wrong.
I'm taking a wild guess. Lawrencinium?
Okay.
Oh, you're really making me work. It's a low budget operation. What can we say? Couldn't afford any more microphones.
I swear I didn't plan it this way.
All right.
So there was 2 people, I think. Yeah.
Are you sure? Okay.
Is it plutonium?
Nope.
Oh man, this is a good one. Please, somebody close.
Alan.
Technetium.
It is technetium.
Bravo, Alan.
In 1937, I believe. Yeah. Okay, so next up, all the way from the computing sciences area, it's Tess Smith.
One of humanity's greatest feats is its increasing ability to understand and manipulate matter. Yet as far as we've come from the Stone Age to the Age of Information, we're far from mastering matter in its most fundamental form, the atom. Like toddlers, puppies, and other small things, atoms are uncooperative. Putting atoms where you want them and then seeing where they actually ended up, it's exhausting and very hard. Because of this, I hung up my lab coat in grad school and decided to take an approach to this using computers. Using existing computational techniques, given an arrangement of atoms, scientists can predict its properties. The question is, how do we find new, useful arrangements of atoms that are worth experimental efforts? Just as written language is not made of all combinations of letters, punctuation, and spaces, atoms do not form random arrangements. They form recurring geometric patterns at many length scales. But the language and the rules of this language that atoms form are very complex and hard to write down. Valid geometries have to satisfy quantum mechanics, and quantum mechanics is a harsh design critic. So I set out to build an algorithm that can learn the language of atoms using a machine learning technique called deep learning. If this program can observe and understand the patterns that we see in experiment, it can learn to combine them in new ways. Deep learning algorithms and the neural networks that they're made of are top performers in translating text and identifying whether an image contains a cat, of course. But existing deep learning techniques don't really understand geometry. If I take, for example, your favorite molecule, let's say it's water for simplicity, and we rotate it, you and I know it's the same thing, but to a neural network, it's an entirely different object. So I had to fix this by building a new type of neural network that can identify the same geometric pattern in any orientation or location from seeing one example. This property turns out to be very useful beyond my original purpose of generating new atomic geometries. Turns out this property makes these networks super able to efficiently emulate expensive physics calculations because these networks obey the same symmetries as physics. Right now, I'm using these networks to compute the quantum mechanical properties of a given arrangement of atoms 6 orders of magnitude faster than before. In the time of this talk, I could have calculated all the supercomputer calculations that I did in my entire PhD. That's pretty cool. Right now, I'm building networks to generate new geometries, new atomic geometries, which may help the notorious challenge of making and measuring materials. And it's my hope, with these tools in hand, we're well on our way to becoming masters of atoms.
Don't leave. Come on. It's trivia time again. Which Berkeley Lab Nobel laureate determined the path of carbon through photosynthesis?
Wasn't it Melvin Calvin?
Sir, you are correct. Melvin Calvin, everybody. Give it up for Mel. And now, from the biosciences area, Feng Chao Song!
Well, Megan and Lauren just gave a great talk showing you microbiome are all around the world and they influence the health of Earth, plant, and actually you as well. However, it is very difficult to understand the function of bacterial communities since it typically contains thousands of species, million cells, and they talk with each other. The interaction determines function. Just like us, actually, in your lab, you either collaborate with each other or, hopefully not, fight for authorship, equipment, cookies. And all their interactions influence your productivity. So people try so hard to study the bacterial interaction by isolating each individual species and pairwisely mix them together and see how they interact. But there are two challenges. First, we are not able to isolate all the species. Second, even if we have all of them, we need to do millions of experiments to test all the interaction. I can tackle this problem by using a very simple idea, bubble. When you pour a little bath soap into water and stir, you get millions of bubbles. Similarly, I make bacterial solution with oil and surfactant and stir. I get millions of liquid bubbles. The bubbles randomly capture some bacteria together and separate them from other bacteria in other bubbles. This allows us to test millions of interactions in parallel without isolating any single species. This is pretty, but there's still a problem. The sizes are not uniform, which means big bubble has more cell, small bubble has less cell. It causes difficulties on testing pairwise interaction because we really need 2 cells in each bubble. To achieve this, I made a microfluidic chip. This is a small chip with a micro-scale channel in it. Using it, I can generate 8 million uniform-sized bubbles like this in an hour. This is just a start. I also designed another chip to merge the bubble with other reagents, then I can detect the interaction in each individual bubble in the whole mix. Because the size of the bubble is very, very small. It's just as tiny as diameter of your hair. So all the bubbles is just in a very small tube, instead of using millions of plastic tubes and tons of medium. The small tube can tell us the interaction and also help us to understand the function of bacteria community. Small bubbles can help with answering big questions. I'm Feng Chao. I'm a bubble player. Thanks.
These trivia questions are hard. Okay, so Berkeley Lab, no stranger to big science, played a key role in the landmark Human Genome Project. The Walnut Creek-based JGI sequenced which 3 human chromosomes? Anyone?
Anyone? Let's see.
Anyone at all?
Astroterry? Oh, man. Get them all right. You have one in like a 23 chance.
5, 16, and 19.
Astrid Terry, everybody.
16 and 19 is right. And next up, a new father from the energy sciences area. It's Wenhouse Sun.
There's something deeply human about exploration. Within each of us, there's a great curiosity to venture into the unknown, to discover something new. There's an unfortunate quote about our generation that says, "We are the middle children of history, born too late to explore the earth, born too early to explore the stars." But actually, there's still a new frontier being explored today, chemical space. Now, chemical space is not really a physical place. meaning we can't just sail there like we would to a new island or continent. Instead, we explore chemical space in a laboratory, mixing together new combinations of elements in a reactor. Sometimes, if we're lucky, these elements will react together to form a new compound. Everyone in this room has benefited from the discovery of new compounds, as these new materials form the basis of safer cars, lighter airplanes, faster computers, and longer-lasting cell phones. Which elements will react together to form a new compound? Actually, it's not so easy to say. We have some ideas and principles to guide us, but they're not perfect. For this reason, chemical exploration is a very risky endeavor. Using modern methods of computational material science, we can today calculate whether or not a new material is stable and therefore synthesizable without even having to step foot in a laboratory. Here at LBNL, I use powerful supercomputers to screen through thousands of hypothetical compounds for new stable materials. Then, just as ancient maps were used to help guide explorers to new lands, we can construct stability maps to help guide chemists in their exploratory synthesis of new compounds. Here, I'm showing a map of the inorganic ternary metal nitrides, a rare but important class of materials. A ternary nitride is composed of 3 elements, metal 1, metal 2, and nitrogen. On this map, the rows are metal 1, and the columns are metal 2, so each square represents a potential ternary nitride compound. On this map, which is a stability map, blue means that that ternary nitride is stable. Green means that it might be stabilizable under special conditions, and red means that it's unstable. In other words, it probably cannot be made. On this map, only 25% of the squares are blue, which means that if I were to randomly mix 2 elements with nitrogen, or in other words, to throw darts randomly at this map, I would only have about a 25% chance of successful nitride synthesis. However, guided by this map, my experimental colleagues have already successfully synthesized 10 out of 10 new ternary nitride materials, a 100% success rate. Beyond the nitrides, there remain many other chemical spaces awaiting experimental discovery. We can apply our approach here to further map these uncharted chemical spaces, providing chemists guidance in their quest to extend the frontier of solid-state chemistry. Thank you very much.
Who wants more trivia? Come on, it's exciting. Let's hear it. Okay, thank you. This is a tough one. Which Berkeley Lab endocrinologist conducted seminal work in isolating and characterizing the progesterone receptor gene in a mouse? This momentous finding transformed our understanding of hormone responsiveness in breast tissue.
We had an early entry here.
Mina Bissell?
Nope.
Shamala Harris?
Shraddha Ravani, you are correct.
Whoa!
The mother of Kamala and Maya Harris. Once again, a representative of biosciences area, Patricia Valdespino.
Hey, Matilda, Matilda, she take me money and run Venezuela.
Have you ever had a pet? This is Matilda. This is Matilda when she was young. She came to my door one day and never left, so we adopted her. Cats are so much fun, so I used to play with her as much as I could. And by doing so, everything happened to me. I got scratched, I found fleas, I read about diseases related to pets. But I learned that no matter I wanted to bond with this kitten, I was actually forced to bond with the kitten and all her associated bugs. And this relies on something we kind of recently understood. We are not alone. Basically, each animal and plant live in association with a unique collection of microbes. We are not individuals, as we used to think. Instead, we are systems, or holobionts, meaning a whole collection of microbes and a host living together in the same unit. In these associations, microbes are at the majority, and until recently, an unseen majority. For example, you can find a couple of millions of microbes in a bucket of seawater, and microbes tenfold the number of human cells in your body. Microbes and hosts manage to walk together through time in a very dynamic and complex equilibrium. If you live in Berkeley, you probably know that having different participants living together in the same house is not easy. And although we can roughly predict the role of microbes in the holobiont or in the environment, basically through bulk sequence analysis, the clarification of their structure, function, or interactions is needed more than ever. This has been a discovery, like a revolution for biology. And in the middle of this era of discovery, here at the Berkeley Lab, you can find exciting strategies to study microbes, such as the synchrotron-based infrared spectroscopy, the strategy I use to explore live cells processes from microscale to nanoscale. Biomolecules are infrared visible, so my job is to integrate this chemical dimension provided by synchrotron technologies with lipidomics, proteomics, and other strategies to try to unravel some of the deepest secrets of the microbial world. In particular, I study the microbes that mediate the transfer of carbon from the atmosphere into timely, stable mineral structures, the carbonates. But this is the way I study microbes here at the Berkeley Lab. But at home, I keep studying them by doing other things, such as taking probiotics, drinking kombucha, or playing with Matilde. Thank you.
And so the end is nigh, and we have another trivia. In 1951, this former lab director won the Nobel Prize in Chemistry. Anyone? You can just start guessing Nobel laureates from Berkeley Lab. Yeah, so we have, are slammers allowed to enter? I don't know.
Like this?
You guys have seen the questions already.
No fair then.
Am I allowed to enter?
Somebody, come on. We only have 13 to choose from.
Someone mentioned him earlier. He was one of the four, I think.
Was it Lawrence?
Nope.
Sorry, James, you can't do it again.
If you might know, you could whisper it to someone next to you and they can answer. So we've got...
Is it Seaborg?
Nope. Nope.
Macmillan? Macmillan?
It's Macmillan. And last, by no means least, from the physical sciences area, it's Maria Zurich.
14 billion years ago, the universe was born in the Big Bang. Ladies, gentlemen, and those beyond the binary. But this what happened in the first seconds, just after it, determined the structure of matter we are all made of. I'm a particle physicist, and in my research, I try to understand how the world around us is made out of its most basic constituents. In the beginning, the universe was hot, dense, and filled only with the most fundamental building blocks of matter called elementary particles. Then, while extending and cooling, the atoms were created. In the center of the simplest hydrogen atom, we can find the tiny proton. And I ask, is it the most fundamental particle? What actually is the proton? Well, the short answer is the proton. It's a mess. It's a complete mess with a complicated internal structure. While the hydrogen atom could be compared to a beautiful, well-organized ballet dance when the electron is dancing elegantly around the proton itself would be a rock concert full of drunk, sweaty people bumping into each other. Proton is full of, as far as we know, elementary particles called quarks and antiquarks and gluons, binding everything together. They are constantly interacting with each other, and they are in constant movement. It's a mess. In my experiment called STAR, I reproduced conditions few seconds just after the Big Bang to look inside the proton. I'm especially curious how its magnetic properties, which determine the stability of matter and lie the foundation of the fields of chemistry and biology, arise out of its messy mosh pit structure. I smashed protons accelerated almost to the speed of light and studied the debris of these collisions in huge particle detectors to ultimately probe the direct interaction between the basic constituents of the proton. And what have I learned? Well, first of all, that nature is quite complicated. We learned that gluons play an extremely important role in describing the magnetic properties of the proton. It's a crucial step in understanding the fundamental structure of matter which makes me, you, and our whole world, but as physicists, it just tickled our curiosity. We are building an even more precise experiment called electron-ion collider to look even deeper into the proton and actually to get closer to answering the question, why does the universe exist as it is? Thank you.
Did you all like that? Yeah?
That was a lot of work.
It really was.
Weeks and weeks, months and months. We
Years
really
and
need to give
years.
it up again for the distinguished dozen. That was nothing short of amazing. Judges, I do not envy your job.
But before we all leave, we have a lot of people to thank that brought this event all together. And when we call you out, we would like you to stand up and wave to everyone so that they know who you are. So first off, our 6 distinguished judges, thank you very much, and you have a hard job ahead of you.
Hey, judges!
And please stand and wave to the audience so they know who you are. So the 6 judges.
Judges, everybody, don't get hurt. Bravo!
Next, we actually had dozens of entries this year. So to get to the final 12, there were a lot of screening judges who had the harder job of selecting the final 12. They were Russell, Ethan, Ali, Mark, Aaliyah, Daniel, Christine, Peter, Pamela, Alan, Valentine, Puya, and Vasilia. So if any of you are here, could you stand up and give us a wave?
All right! Bravo!
There were communications and artwork who made these beautiful brochures for you all and put together the slides and did all of the PR that you've seen all around the lab for the last few weeks. So a big hand up to Massey, David, Diane, at Creative Services as well, who were very much involved in putting this all together.
Creative Services, yeah.
Of course, you wouldn't be able to hear us or see us without the AV team, so they're up in the booth. Thank you very much. And And anyone else you can think of?
I think have we forgotten somebody? Somebody really important that this couldn't even happen without that one individual.
I don't know. Sorry.
Didn't you practice? What? You've already won, Alan. Sorry. I know. It's Meg Rodriguez. Stand up, Meg. Hey! Seriously, short of making all the oxygen and nitrogen in the room, she did everything. Seriously.
She was here last night making Star Wars videos until 9 p.m., so. Okay.
Okay.
So without that, we'll get the final 12 people.
Oh, right. I just wanted to plant this in your mind. Spoiler alert.
So one more thing. Meg, we want you to come down to the front, please.
Every year. Yay, Meg.
Thank you very much.
Help yourself. You upholstered it last night too, I think. Did everything, this woman, Meg Rodriguez. All right. If you can, stick around for a wonderful reception afterwards in the patio outside the cafeteria. And definitely want you to vote for your favorite, and it's going to be tough. What else we got?
When do we know when the winner is?
Well, I would say the winner will be at 4:30, so that means there'll be plenty of time to enjoy the reception in advance of that announcement. So those of you online, please stick with us.
If you follow Berkeley Lab Postdoc on Facebook, they're going to live stream Facebook. However, I'm not the social media expert.
Like we didn't know. And now, finally, we'd like to invite the finalists, those distinguished dozen, to join us down here on the stage and take their vows. Woo. Vows even. Bravo! All right, then. Thank you very much. But wait, there's more.
If the screening judges could also see me before you leave, but everyone else, if you can help us celebrate and cap off National Postdoc Appreciation Week up at the patio, we would love to have you walk very slow. There was a BART delay, so our bartender's still coming, so walk very slow. 4 o'clock means 4 o'clock, okay? Thank you.
Okay, we've stalled long enough. Thank you very much, everybody. No recordable injuries. Thank you, finalists. Good luck to you all.
Well, first off, thank you all for coming and for sticking around to wait for the winner. Without too much delay, we'll first announce the people that you chose, the person that you chose to be the winner, so the People's Choice Award. And I'm really happy to announce that the People's Choice winner is, from Computing Sciences, Tess Smith.
Congratulations, Tess. Now for the second runner-up. From Computing Sciences, Bashir Mohammed!
Are you all tired? Yeah? So they said I have 3 minutes to announce the winner. No, it's okay. So, without much more delay, I have to pass over my crown, which I've had since this morning. But I was kind of looking forward to wearing it around the foundry for a while, so I'm going to have to pass it on. But the good news is that it's staying in the energy sciences area. The winner is Wen Hao. So thanks everyone, please enjoy the reception and see you next year.